DNA Depot presents Bioremediation by Oil-Eating Bacteria, Kit DD302. Bioremediation is the process whereby living organisms are used to degrade contaminants in the environment and to make it safe for other living organisms. In this experiment, you will discover how oil-eating bacteria are used to clean up oil spills and how components of oil are used as a source of energy for bacteria. The experiment is designed for 6 student experiments to be performed 3 at a time. Your kit should include oil-eating microbes and stabilizer, litmus paper, 3 plastic jars and foam stopper, 6 transfer pipettes, a plastic spoon, and control oil sample. You will also need cooking oils such as vegetable, canola, or corn, various chemical oils such as motor oil, engine oil, lubricating oil, tap water, vinegar, baking soda, salt, gloves and goggles, and a lab notebook. Experiment procedure. 3 experiments can be tested at a time. This procedure gives you directions to test 1 oil sample and 2 control samples. 1. Select an oil sample from cooking oils or motor oils such as engine, transmission, or brake fluid oils. 2. Clean and dry the plastic jars before using. 3. You should have 1 jar for the control oil, the selected oil, and 1 for the negative control, no oil. Label each jar accordingly. 4. Place paper towels or newspaper over your work area. 5. On the outside of the jar, use a ruler to measure from the bottom of the jar and mark with a permanent marker a line at 2, 2 1/4, and 3 inches. 6. Fill all 3 jars with room temperature tap water to the 2-inch mark. 7. To the negative control jar, add more water to the 2 1/4 mark. 8. To the selected oil sample jar, use a transfer pipette and add drop-wise the oil sample to the 2 1/4 mark. 9. Repeat step 8 with the control oil provided. 10. Use the disposable plastic spoon to carefully add 1/4 teaspoon of oil-eating bacterial mix to all the samples. Just sprinkle the bacterial mix over the water. Place a sponge stopper in the plastic jars. Place the 3 plastic jars in a larger jar such as a pickle jar to prevent the jars from tipping over, and place in an area where they will not be disturbed. 12. You can take a picture of the jars at day 1 for comparison with later observations. Take pictures every 48 hours. At the completion of this set of experiments, the jars can be disinfected and cleaned, and the experiment can be repeated with 3 more oil samples. In this experiment, you've discovered a form of bioremediation. There are other organisms that are also used to get rid of contaminants in our environment. The new green revolution in the USA and the world emphasizes that it is better to eliminate the use of synthetic chemicals for cleaning our environment.